Okay, that was a Snapchat that I received from a friend that studies bacteria. So perhaps by the end of this lecture, we'll be able to understand what was going on in that video. This lecture is intended to help you through your study of bacteria. Since I'm not here today, you'll be listening to the video, taking notes in your notebook, and you should have your textbook nearby as well. If at any point during the video you need to pause it, to copy down a note, or to grab your textbook, that's fine. The slides that have stars on them are the ones that you are intended to copy down. Let's get started. How many bacteria do you think are in your mouth right now? Maybe a few, maybe some of us forgot to brush our teeth this morning, right? Actually, in each one of our mouths, there's more bacteria than there are humans in the world. Our mouth contains billions of bacteria, which, oddly enough, is a sign of health rather than dirtiness. So what are bacteria? Bacteria are prokaryotes. Remember, prokaryotic cells do not have membrane-bound organelles. Instead, their genetic material floats around inside of the cytoplasm. Bacteria are unicellular, meaning they're single-celled organisms. They basically contain DNA, or genetic material, cytoplasm, ribosomes, which translate the DNA into proteins, a cell membrane surrounding the inside, a cell wall, providing extra protection and support. And bacteria have these little tails called flagella, which help the bacteria move. At this point, I'd like you to go to page 218 or 219 in your textbook and draw the image. Pause the video as you do so. How do bacteria get their energy? Bacteria can be autotrophs or heterotrophs. Recall that an autotroph is an organism that's able to produce its own food, kind of like a plant gets its energy from the sun. Heterotrophs rely on other organisms for food. So bacteria can either be autotrophs, they can carry out photosynthesis like plants, or they can get energy from other chemicals, or they can be heterotrophs and rely on other organisms. Bacteria come in many shapes. They come in spheres, like Coccobacillus. They can come in rods, kind of look like a pill, like Bacillus or Vibrio. Or bacteria can look kind of like a spiral, like a spirochete. Just draw those shapes. You don't need to name them. Bacteria can reproduce in two ways. They can reproduce asexually by binary fission. Now, in asexual reproduction, there's only one parent, and the offspring is identical to that parent. It's basically a process of cloning. We call this process binary fission. In binary fission, one cell splits or divides to form two identical cells. This can happen as often as every 20 minutes. To give you an idea of how quickly bacteria can build up, we can watch this quick video.
Produce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Eek. So, binary fission, or bacterial asexual reproduction, occurs very quickly. Binary fission, asexual reproduction, the offspring are identical to the parents. Bacteria can also reproduce sexually, and that involves two parents. When two parents are involved, the offspring is no longer identical to either one of them. You can imagine you look a little bit like both of your parents, but not exactly like either one. Sexual reproduction in bacteria is called conjugation. And in conjugation, one bacterial cell basically just shares its genetic material with another. So the genetic material can be transferred between bacterial cells, and if you gain new genetic material, you basically become a new individual. So asexual reproduction is binary fission, sexual reproduction is conjugation. Bacteria play many important roles in our environment. Number one is oxygen production. Bacteria actually produce more oxygen than all plants combined. Recall that we said that some bacteria can be autotrophs. Well, when bacteria photosynthesize, they release oxygen. Bacteria also play an important role in food production. First, we of course want to get rid of the bad bacteria in a process called pasteurization. So maybe you've heard of pasteurized milk. Pasteurized food has been heated to kill off the bad bacteria. However, there are actually bacteria that play a very positive role in many of our foods. If you go to pages 222 and 223, you can find and list a few of the foods that we use bacteria for. Pause the video while you do that. Another important role that bacteria plays for our environment is environmental recycling. Bacteria are organisms called decomposers. Decomposers basically break down dead organisms. If you can imagine a rainforest, right? In a rainforest, animals live and die, trees fall, leaves fall, and if none of those dead organisms, those dead trees, those dead leaves, those dead animals were removed, it would pile up very quickly and suffocate the rainforest. So what bacteria sometimes do is basically eat or remove those dead organisms so that they can be recycled back into the environment and reused. So decomposers break down dead organisms. They recycle nutrients. Bacteria can also be important for environmental cleanup. Maybe you've heard of some major oil spills where thick black oil spills out into the ocean and kills many different birds and fish. But bacteria can actually help break down that oil so that it's not quite so damaging to the environment. Bacteria play an important role in health and medicine. There's many good bacteria in our body. Like we heard in the beginning, there's tons of bacteria in our mouth, even more in our gut and stomach. So the bacteria in our stomach help us digest our food. They help keep our digestive tract healthy. Bacteria are very important for our body to function normally. Perhaps you may know someone that has diabetes. Individuals with diabetes are not able to produce insulin. Insulin helps manage their blood sugar. So for those people who cannot produce their own insulin, bacteria can actually be engineered or created to make it. And then those individuals can use the insulin from the bacteria. And of course, there are the pathogenic or the infectious bacteria that cause diseases. So path, remember, means disease. Pathogenic is disease causing. So maybe you've heard of or had a few of these, anything from an ear infection, strep throat, staph, pneumonia, salmonella, E. coli, tuberculosis, or the bubonic plague. So take a moment to write those down. If you recall, is the flu or the cold caused by bacteria? Uh-oh. That doesn't mean you can go. No, the flu and the cold are caused by viruses. So how do we get rid of bacteria? Or how do we get rid of bad bacteria? We can take antibiotics. Antibiotics are medicines that stop the growth of or destroy bacteria. Anti, remember, means against. Bio means life. So antibiotics are against the life of bacteria. Antibiotics do nothing for viruses. If you have the flu, if you have the cold, You cannot take antibiotics for help. Antibiotics only act against bacteria. So how were antibiotics first discovered? It's a funny story.
The year is 1914. The European subcontinent is embroiled in bloody trench warfare. Soldiers on all sides are victims to artillery fire, mustard gas, and new automatic rifles. Their biggest enemy, however, is infection. Of those wounded in battle, few will survive even their minor injuries. Field hospitals populate the front lines, with young doctors and nurses working day and night to stem the stealthy killer. Their main defense is antiseptic, which proves just as lethal to the patients as no treatment at all. One frustrated young doctor called Alexander Fleming is unwilling to accept the inefficacy of current medicine to rescue the wounded in battle. He would later dedicate his life to understanding and fighting infections, going on to pioneer one of the most influential medical discoveries of the 20th century. A discovery which Time magazine claims has altered the course of history. Fleming was born in Ayrshire, Scotland, to a farming family. He performed well at school, later moving to London to attend the Royal Polytechnic Institution, after which he took to working in a local shipping office. Fleming would work there for a subsequent 4 years, until receiving a fortuitous inheritance from his uncle that would see him through St. Mary's Hospital Medical School. It was this medical training which would later take Fleming to the front lines as a captain in the Royal Army Medical Corps, where he would gain first-hand knowledge of the perils of treating soldiers in battle. Before the outbreak of the First World War, Fleming had become assistant bacteriologist to Sir Almroth Wright, a pre-eminent scientist in the fields of bacteriology and immunology. His stay at the university was extended only at the insistence of the president of its rifling club, of which Fleming was a member. To remain active, Fleming took up the research position under Almroth in the immunology department. Unfortunately, Fleming's work was derailed when Britain joined the war effort on the 4th of August in 1914. By the conflict's close, Fleming had witnessed the need for a replacement to the ineffective and deadly antiseptics in use. Upon his return from the service, he resumed his position at St. Mary's, and with strong support from Wright, he dedicated much of his time to studying the effects of antiseptics and their unintended lethality in the war effort. A major breakthrough in his work came in the form of the discovery of lysozyme in human mucus. This naturally occurring agent protects the human immune system from certain common bacteria. By 1928, Fleming had been appointed Professor of Bacteriology at the University of London, leading research on common bacteria such as the staphylococci, which may be found on household surfaces. Ironically, it was the somewhat undesirable trait of Fleming's untidiness which would ultimately modernize the human effort against disease. Upon his return from a month-long holiday, Fleming found in his lab a petri dish upon which a fungus had grown. The dish contained a Staphylococcus culture, but around the mold growth, he found that all the bacteria had died off. His former assistant Merlin Price prompted Fleming to investigate the growth, reminding him of the lysozyme discovery. Of the occasion, Fleming later said, "When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all of medicine by discovering the world's first antibiotic or bacteria killer. But I suppose that was exactly what I did."
Video is about to cut me off, but we'll discuss this lecture tomorrow in class. Please complete the homework as instructed on the plan sheet.